Thank you so much for having me virtually. I'm live casting from Rochester, New York. I'm a PhD student in Nancy Chen's lab. And today I just wanted to talk a little bit about this large-scale population genetic data set for the Florida scrub jay that the lab is currently generating, and a little sprinkle of my future plans with this data set. Cool. So how do small populations evolve in nature? This is a big open question, both in conservation biology and just evolutionary biology in general, I would say. And so some sub-questions that we can maybe ask under this topic are, how are units of genetic information distributed across time and space? What's the genetic landscape of inbreeding? And how is that genetic landscape linked to these observed negative fitness consequences that we see? And then more genomics related questions we can ask are like, what mutation rates do we observe, and recombination landscapes can we observe? And so these are all projects that members of the Chen Lab here at Rochester are interested in pursuing. And obviously, I don't have to convince anyone in this group that the Florida scrub jay is an amazing and excellent and beautiful system to begin exploring these questions, given the massive wealth of data that is individually based, and we have life history data and genetic data collected for these jays, given the tons of work of people that have put in over the past 50 years and beyond. And particularly, this data is very rich for the Archbold Biological Station population. And I want to highlight here blood samples. So because we're like an evolutionary genetics group of folks, we're interested in these blood samples. So how do we actually get from blood samples to high quality DNA data? And we need this high quality DNA data to answer these questions that I've posed here, and hopefully questions beyond that. And so what the Chen Lab is currently doing to try and answer these questions is actually generating from those blood samples whole genome sequence data for the around 3,800 jays that have been present in the Archbold Biological Station population from 1999 to 2021. And so like I alluded to before, how do we actually get from blood samples that folks have collected in the field to actual informative genetic sequence data that we can like look at on a computer? And so I won't go into the details of this pipeline that I just put on the screen here, but it involves a ton of lab work in sorting these 3,800 blood samples, transferring and extracting DNA, making sure it's ready and concentrated enough to ship out for sequencing, and then dealing with what happens when your samples aren't up to the quality standard and having to redo this process over and over for these thousands of samples. And then once you actually have raw sequence data, there's this huge computational component where you receive that raw data, you have to clean it up, and you generate these large files called VCFs. I don't know if you can see my mouse, but on the right-hand side of the screen, you'll see the acronym VCF. And these are just summaries of that genetic variation across all these individuals. And this whole time, you need to back up your files, make sure everything's running correctly and not failing mysteriously in the middle of the night and you don't catch it. And you also have to keep track of where everything lives. So we're dealing with thousands of terabytes of data. And so also data storage and managing the computational workload on a shared computer is also a big task. Okay. And so last time I gave a talk at Archbold, which I think was earlier this year in the spring, I mentioned that I was working on one of the first pieces of data that we need for this data set, which is actually a high quality reference genome. So for folks who don't know, a reference genome is the genome from a single individual that serves as a point of comparison. So when we actually align the 3,800 genomes of all the other Jays to this reference genome, it actually lets us characterize this genetic variation present across all the individuals in a standardized way. And I'm actually super happy to report that I recently finished assembling this genome, and it's actually now publicly available on NCBI and has been labeled by NCBI as the new publicly available reference for the Florida scrub jay, which is super exciting. This has been sort of what I've been really pushing to get out over the summer. And so the paper also just got accepted to G3. I have like a little citation in the bottom right corner. So it just got accepted and should be published soon. And very exciting. And some fun facts about this genome and why it's an improvement on the other genomes that have been published in the past is that we added 260 megabases of sequence, which is a lot of sequence, because we used long read sequencing techniques. So that added a lot of hard to sequence elements of the genome, like repetitive regions. We also, in this genome assembly, identified 4 new chromosomes to the Florida scrub jay, including the W, because the previous genomes were all male individual scrub jays, and this is the first female. So we actually have the W chromosome now. And like I just mentioned, we have better resolved repetitive regions and those sort of mysterious regions of the genome. And also, along with the genome assembly, we were able to publish the first linkage map for the Florida scrub jay that Nancy has been working tirelessly on for quite a while. And so all of that is available to the public. And if you have any questions about it, please let me know. But yeah, I hope this genome will be a super useful tool for anyone wanting to do scrub jay genetics or just bird genetics in general. Okay, so back to this previous slide of our goals and our little pipeline here. I put a little tick on create reference genome because we have ticked that off our list. And so now we have to get DNA from blood, and we have to do this for almost 4,000 samples. And so I just wanted to give a little fun slide of DNA extraction adventures. So like I sort of said in the beginning, this involves sorting through, so if you look in the middle top row, there's an image of, I don't know, that's maybe 3 or 5, 6, like almost 70, 80 blood boxes. And within each of those boxes, there is a single tube of blood, and there could be 100 tubes in that box. And so this adventure has entailed sorting through all those, going into the wet lab, extracting DNA individually from each of those tubes or using plate extractions, has also involved scary thawing of the fridge and making sure all the DNA is safe. This is a picture of Jeremy on the right, who's going to be giving a talk after me, sending our samples off to sequencing. And I've also included images of other folks who have sequenced some of this data as well. Summary, it was a lot of work and a lot of hard work from a lot of folks. And then last thing is that this image in the bottom of like the stack of papers. So each paper is tracking the samples that we're shipping, and each paper is a box. And so that stack of papers is maybe like 1.5 inches thick. So that gives you an example of how many boxes of samples we actually shipped to get sequenced. So big endeavor, but really exciting that it's sort of we've like reached the end of this part of the pipeline through the hard work of a lot of people. And I just wanted to give a shout out to that part of the process. Okay, so we have extracted DNA from these 3,800 samples. And now I want to talk a little bit about the sequencing strategy that we're employing. Okay, so we are sequencing about 600 Jays to high coverage and around 3,200 Jays to low coverage. And so what is coverage? So coverage refers to the depth of sequencing, or like how many times each base in the genome is read. So for our high coverage individuals, each like location, each base is read about 20 times. So high coverage gives you much higher confidence in the accuracy of your sequence. Like you're like, okay, I know that there's a T at that location. Like, great. But the downside is that it is much more costly to do this high coverage sequencing. In contrast, just the bottom image there, low coverage sequencing means you only read that base pair maybe once or twice. And so there, like I mentioned, there are obviously cost restrictions to generating, a data set, a whole genome data set of almost 4,000 bird genomes. So the majority of our samples are sequenced to low or 2x coverage because of this restraint. However, we can actually leverage the amazing pedigree that we have for these birds to actually help maximize our sequencing scheme. So the question is like, okay, you can sequence to high and low, but how do you actually choose which J's are high versus low coverage? So on the right hand side here is just a toy pedigree that isn't like a real pedigree or related to any data. But if you just take a quick glance at this pedigree, you can imagine that there are individuals in it that are more informative than others. So in other words, like you can imagine that there are these nodes at the pedigree that have individuals that are related to a large amount of other individuals in the family. So given that, there could be chunks of their genome that are more represented and exist in more copies down the pedigree in relation to other individuals. So actually, I think, okay, cool. So I've starred here some examples of individuals that might be the case. And so like I said before, you can actually use the pedigree to help us strategically select specific individuals to sequence to high coverage, maximizing genetic information. And so you can kind of get away with like, at the bottom of this pedigree, you might be sequencing all these individuals to like 2x coverage, but you can increase that coverage later in the bioinformatics processing because you've sequenced their parents really well. So that's the general idea there. And I think I said that again. Leverage greater sequence confidence of these start individuals to fill in the gaps in their relatives' genomes. Okay, and so I actually have this lovely graph of our current sequencing progress of the 3,800 ABSJs. So I'll explain this figure here. Each panel shows the number of J's on the y-axis present in ABS across time. So year is on the x-axis. And these panels are split by social class assignment. So you have breeder, non-breeder, and juveniles. And then the y-axis is split into colors based on the current sequencing status. So you have, or in orange, you have high coverage individuals, which means that we currently have on our computers like the 20x coverage sequence data for those individuals. And then low is the same thing, but we currently have 2x coverage sequencing data for those individuals. In purple, we have waiting for sequence data, which we're currently waiting to receive that sequence data. And then no-seq data is that individual has not been sequenced. And so as you can sort of see over time, we've made a lot of progress, which is very exciting, especially over the last like 8 months or so. And the biggest batch we're waiting for is we're mostly waiting for the last batch of like 2021 nestlings to arrive. So this big bar, I don't know if you can see my mouse, but on the right hand side of the juvenile plot for 2021, we see this giant chunk of purple that we're waiting to receive. Okay, and so awesome. So even though we're still waiting on sequence data, so I put a little clock here, we can actually start processing the sequence data that we already have through the rest of the bioinformatics pipeline. So again, like I said at the start of this talk, the raw data needs to be cleaned up, aligned to the reference genome, and processed further until it reaches like its final file form that we can use for like population genetic analyses in the future. And so just as a super quick, I think, update on where I am there, I've processed about 500 of the 600 high coverage samples through the joint variant discovery phase, which is the generate VCF file plus filter part of this pipeline. And then I've processed a little more than 2,000 low coverage samples through just that very first read quality control and trimming step. So obviously we still have a long way to go, but I feel like we've really made a lot of progress. And I'm really working on making this sort of a more streamlined and automated thing that I can just press go on, keep track of, and push these samples through in a timely manner. So that's sort of my task right now. And then I just wanted to give a little bit of information on what I personally hope to do with this giant data set once it's finished. And so I'm personally really interested in using this data to explore the underlying genetics of inbreeding depression. So we know from previous work done by Nancy and others in the ABS population that inbreeding levels have been increasing and fitness has been decreasing in the population over time. And so the really exciting thing that we can actually do with whole genome data is we can actually characterize inbreeding at the genetic level by looking for these runs of homozygosity. And these are just stretches of an individual's genome that are completely homozygous due to the parents passing on identical haplotypes, identical stretches of the genome to their offspring. And so I think we can ask some really interesting and really fundamental questions just about like the genetic composition of small natural populations that probably have some inbreeding going on. So how are these runs of homozygosity distributed across time and across individuals? Can we actually see inbreeding levels increasing over time in the genome? And then sort of on top of that, we can also ask whether individuals with more runs of homozygosity are less fit, right? Because like theory predicts that the more homozygous you are, the greater the chance that these recessive harmful alleles are expressed. And so you're predicted to be less fit. But this has actually rarely been tested in natural populations because basically no one or very few people have sort of the life history data plus the pedigree, plus the genetic resources to actually explore this question. And so my little sort of summary of this is that we're really uniquely posed to answer this question of like, we know inbreeding is bad. We know inbreeding is bad in small populations under threat, right? But why is it bad, right? And so if we know this at like the genetic level, maybe we can sort of work backwards and make more informed conservation management decisions in the future. And so that's what I hope to do with this data set once it's made. And I don't know how long that took me, but I would like to acknowledge all the folks at the University of Rochester and my PI Nancy for all their hard work during the wet lab stages and beyond. Also more collaborators at Cornell and beyond, and also everyone at Archbold for collecting this amazing data and for doing the actual hard work to maintain this amazing system. And I would be happy to take any questions. Thank you.
Hey, this is Sahas here. I hope you could at least hear a little bit of the loud applause you got.
Thank you.
I'm going to be here to relay any questions that folks might have. I'll just repeat the question, so it's next to the mic so Faye can hear it. Yes, right.
Please tell us. Electricity and freezers and
Fred wants to know how do you make sure that your freezers don't die on you, or what contingencies you have for that?
Oh my gosh,
or anything else.
What an amazing question. Yeah, so basically it's been a big adventure of realizing that every time you open the freezer, it lets in a little bit of humidity and moisture, right? And so over time, ice is going to crystallize in the freezer. And so over the years, we've learned that we really need to defrost the freezer like every 6 months or so. And we actually, so we have a -20 freezer that we keep a lot of DNA samples in. And then we have a -80 centigrade freezer that we do long-term storage in. And so twice a year, hopefully, this is what we're trying to do. You like move all the DNA samples to the -80 and you defrost the freezer so that there's no scary, oh no, the freezer wasn't able to close and it was left overnight and then all the samples thawed. Like that is what we're trying to avoid. So it's just, you know, regular wet lab maintenance, and also if there's a power outage, making sure that we're communicating with like building staff and everyone in the lab to make sure that it's all good. But so far we haven't had, I mean, Jeremy can correct me because he's been in the lab longer than me, but I don't think we've had like any huge disasters or anything. I did have to mop the floor a couple times, but like happy to do that if it saves the samples.
Let's do maybe one more question. How about?
The sequencing data that he's waiting on, is that all low or high or a combination of both in the same proportions roughly that you have?
Yeah, so the question, Faye, is that the sequencing data that you're waiting on, is that all high coverage, low coverage, a mix?
Yeah, so
what are we waiting?
Yeah, so most of what we're waiting on is, I believe, low coverage. It's about like 800 low coverage, and the numbers always fluctuate because what happens is we send stuff for sequencing and we're like, cross our fingers, everything's gonna go great. And then they always tell us, okay, here are like, you know, 5% of your samples didn't pass quality, you need to send more DNA. And so it's just sort of this constant like cyclic process of like making sure, like retrying samples. So yeah, mostly low coverage. And then I want to say like maybe less than 50 high coverage that we're waiting on, but we're expecting it in like the next month or so. Again, Jeremy can correct me if I'm wrong, but yeah.
Well, thank you so much, Faye. And speaking of Jeremy, let's